everyone, welcome to the 1000 MCQ series of Food Safety Officer examination. This is our exclusive course designed for FSO in which you will get more than 10,000 MCQs at just rupees 499. Coming to the first question of the day, who is credited with coining the term chromatography and performing the first chromatographic separation? Yes, it is Martin Sinch, Michael Twist, Archer Martin, or Richard Sinch. And the answer is Michael Twist. See, Martin and Sinch, that is Archer Martin and Richard Sinch, had developed partition chromatography. They had together developed this and won the Nobel Prize in Chemistry. And their work was the separation of amino acids, while Martin Twist was considered as the father of chromatography. The scientist had separated plant pigments. Coming to the next question, the fundamental principle underlying chromatographic separation is it differences in the molecular weight, differences in the boiling point, differences in the affinity for the stationary phase, or the differences in solubility in the mobile phase? Yes, it is the differences in the affinity towards the stationary phase. Next question is, which chromatographic techniques involve liquid stationary phase held on an inert solid support and a gas mobile phase? Here we have to note two things. What is a stationary phase? What is a mobile phase? Stationary phase is liquid and mobile phase is gaseous. So the options are gas solid chromatography, liquid solid chromatography, gas liquid chromatography, or liquid liquid chromatography. So the answer will be gas liquid chromatography. Right. Now, if we see the classification of chromatographic techniques, we can divide it based on three main factors. First one is based upon the phase state. Second one is based upon the operational technique. And third one is based upon the separation mechanism. Now, coming to the phase state, it is mainly divided into two types, LC and GC. That is liquid chromatography and gas chromatography. In liquid chromatography can be further divided into liquid solid chromatography and liquid liquid chromatography, while gas chromatography is divided into gas solid chromatography and gas liquid chromatography. Coming to the operational techniques, it is mainly divided into two types, that is column chromatography and planar chromatography. In column chromatography, we are using a column which is packed with the stationary phase. Right. So separation in a column which is packed with a stationary phase takes place. In planar chromatography, a flat surface we are using, such as in TLC. Right. So the separation is on a flat surface, such as Draper chromatography as well as thin layer chromatography. Now, coming to the separation mechanism, it is mainly two types, adsorption or partition. Adsorption chromatography means it is getting adsorbed onto a surface. The separation is based on the adsorption onto a solid stationary phase. While in partition chromatography, partitioning takes place between the two phases. That is, separation is based upon the partitioning between the liquid stationary phase and the mobile phase. That is partition chromatography. Now, which of the following is separated through column chromatography? Is it chlorophyll and carotenoids, inorganic cations or complexes, sugar derivatives, or amino acids which are formed by hydrolysis of protein molecule? Yes, the answer is chlorophyll and carotenoids. Let's see the other options. They have given inorganic cations and complexes. So the cation consists of ion or charge. So here we will be using ion exchange chromatography. Sugar derivative. For sugar derivative, we use size exclusion chromatography or HPLC. That is high performance liquid chromatography. For amino acids which are derived from the hydrolysis of protein, we can use ion exchange because amino acids contain charge, or we can use TLC, that is thin layer chromatography. What is this column chromatography? Column chromatography is basically a separation technique. Here we are separating a mixtures on the basis of what? On the basis of differential adsorption to a stationary phase. Right. So in column chromatography, if we see, there is a particular column is used. Right. Inside this particular column, it will be filled with stationary phase, and we will administer or we will inject the mixture or the sample to this stationary phase with the help of a mobile phase. So there are mainly two phases, the stationary phase and the mobile phase. So the stationary phase is a solid adsorbent such as silica gel or alumina which is packed inside this column. Now what flows through this column, that is a mobile phase. So it can be a liquid solid that carries the mixture. Mixture means the mixture that we want to separate through this column packed with the stationary phase. Right. So that is the basic setup of a column chromatography. Now what is the principle? First, the mixture is applied to the top of the column. The mixture or the sample that we want to administer or separate will be administered at the top of the column, and as the solvent moves, there will be separation taking place. How? The component with higher affinity will stick on with that stationary phase and it will move very slowly, while the components which are having weaker affinity, which are not very easily bound, they will slip out easily and move faster through the stationary phase. This is how separation takes place. Now, what are the applications of column chromatography? This can be used for the purification of compound, analysis of mixtures, environmental analysis, separation of isomers, and drug discovery and development. Now, coming to the next question. To improve the resolution in a chromatographic separation, you must decrease the number of theoretical plates in the column, increase the height of the theoretical plate in the column, increase both the number and height of the theoretical plate in the column, or increase the number of theoretical plate in the column. Yes, the answer is we should increase the number of theoretical plate in the column. And if a question comes like, what about the height? Height means we should decrease the height of the theoretical column. Right. So here the best answer is so as to increase the number of theoretical plates in the column. Right. Now, coming to the next question. Vitamin D is detected by TLC by using which component? That is it iodine, antimony trichloride, acridine orange, or ninhydrin? Yes, the answer is antimony trichloride. See, antimony trichloride can be used for the identification of flavonoids, vitamin A and D, carotenoids, steroids, and terpenes. While what about iodine vapor? Iodine vapor is a universal reagent. Okay, so it can be a relatively unspecific universal reagent for many types of organic compounds. Coming to ninhydrin. Ninhydrin is for amino acids or amine sugars. Right. So ninhydrin can be used for the identification of amines and amino sugars. Ninhydrin or cadmium acetate. If a question comes like that, cadmium acetate can be used for the identification of amino acids and heterocyclic amines. Coming to ninhydrin, pyridine or glacial acetic acid, it is used for the identification of peptides. Coming to acridine orange, it is for DNA and RNA. Anisodine phthalate. Anisodine phthalate is for carbohydrates as well as reducing sugars. Now, bromothiamol blue is for fat. Right, fat in the sense lipids and phospholipids. Now, dichlorofluorescent. These are for sugars. So, dichlorofluorescent is for sweeteners such as saccharin and cyclamates. Now, coming to the next question. The most widely used HPLC detector which is suitable for gradient elution is it refractive index detector, UV detector, electrochemical detector, or fluorescence detector? So the answer is UV detector. Right. So coming to this HPLC, in the question it is asked which is the detector used in HPLC. Right. So coming to HPLC, what is HPLC? It is high performance liquid chromatography in which we are using high pressure. Right. So the separation of the compounds is by using pressurized liquid through a column. Now, there are different types of chromatography. First one is partition. Partition chromatography is based upon the polarity of the compounds. Coming to adsorption chromatography. Adsorption means on the flat surface there will be surface adsorption. This is how adsorption chromatography works. Ion exchange chromatography is mainly for ionic interaction and the molecule or compound which are having charges. Coming to size exclusion, these are mainly by separation is mainly by the differences in the molecular size of the compound. Coming to the affinity, affinity is for specific binding in which we will be using many type of chiral compounds also. Okay, that is the types of chromatography. Coming to elution, elution can be of two types, isocratic elution or gradient elution. In isocratic elution, it is a constant solvent. It will be used for elution process. While in gradient elution, there will be change in the solvent that we are using. Right? Either there can be change in the ratio of the solvent, or it can be a different solvent itself. Now, what are the detectors which are used? It is UV detector, fluorescent detector, conductivity detector, as well as MS. Sometimes HPLC is combined with a spectrometry, that is mass spectrometry, for the identification of unknown compounds. Right. Now, coming to the HPLC mode, there are different modes for the HPLC. Normal phase means it is consisting of a polar stationary phase. Reverse phase means it consists of a non-polar stationary phase. Right, that is normal phase and reverse phase. Coming to ion exchange, it is mainly for ionic compound. Size exclusion means it is for change or difference in the size, mainly for larger molecules. Now coming to affinity and chiral, these are specific separations based upon the specific binding of these compounds. Now coming to the suitability. Suitability means in non-polar solute, the main mechanisms that act is adsorption and gel permeation. In ionic solvents, if you are using, it will ion exchange and gel filtration. Coming to polar solvents, it will be partition. Partition chromatography will be used. Right. Now, which is the best match between detectors used in GC, that is gas chromatography, and the compounds to which they are sensitive? Right. There are different types of detectors that they have given. That is thermal conductivity detector, flame ionization detector, electron capture detector, nitrogen phosphorus detector. Now let's start with the last one itself. Nitrogen phosphorus detector. This is used for what? For nitrogen and phosphorus containing compound. Electron capture detector. This is used for what? Electron withdrawing species. Flame ionization detector. This is for organic compounds. And thermal conductivity detector is for all type of species. Right. So this will be the matching. Now, if we see each one in detail, thermal conductivity detector, that is TCD, it is a non-destructive type of detector which responds to the change in the thermal conductivity. Right. It is less sensitive in nature and detect most of the inorganic gases such as the hydrogen, oxygen, nitrogen, carbon monoxide, carbon dioxide, etc. Coming to flame ionization detector, it is a destructive detector which responds to the ionization of these compounds. See, in flame ionization detector, we are using a particular flame, and the flame that is used is hydrogen flame. These are very much sensitive to organic compounds. They are mainly used for hydrocarbons, right? For example, petroleum, or for environmental analysis. Coming to electron capture detector, that is the ECD detector, it is a destructive type of detector where we are checking the electronegativity. So most probably the compounds that we are using will be negatively charged. For example, halogens or nitro groups. These are used for the detection and analysis of pesticides, herbicides, as well as halogenated compounds. Now, nitrogen phosphorus detector, that is NPD detector. It is a destructive detector. It is very specific for nitrogen as well as phosphorus containing compounds. So this is mainly used for drugs, pesticides, food additives, etc. That is NPD or nitrogen phosphorus detector. Coming to the flame photometric detector or FPD, it is a destructive detector where we will be specifically analyzing sulfur and phosphorus containing compounds. It is used for pesticide, herbicides, as well as sulfur containing petroleum products. Coming to the photoionization detector, also known as PID detector or photoionization detector, it is a non-destructive type of detector, mainly like specific sensitivities towards aromatic compound as well as unsaturated compound. Here we have to note one thing. PID detector, that is photoionization detector, is mainly used for VOCs. VOC means volatile organic compounds. Because of this, they are also used for the analysis of air pollutants. Now, next one is electrolytic conductivity detector, ECD detector. Right, it is not electron capture detector, it is electrolytic conductivity detector. So this is also a destructive type of detector and it is mainly selective for halogenated compounds. Right, so here we are analyzing pesticide, herbicide as well as halogenated chemicals. So these are the various type of detectors. Now coming to the next question, an unsuitable carrier gas in gas chromatography. An unsuitable carrier gas. See, whether it is helium, hydrogen, nitrogen or oxygen. Yes, The answer will be oxygen. Why? Because of its reactivity. Carrier gas are usually noble or ideal in nature. They are non-reactive, but in oxygen it is highly reactive. Okay. So coming to helium, helium is generally used as a carrier gas, as a makeup gas, but it is very expensive. Coming to nitrogen, nitrogen is also used as a carrier gas and a makeup gas. It is very cheap, but the main problem is it is not good for capillary columns because it takes a long run time. Coming to oxygen, oxygen is considered as a combustion gas, mainly for FIDs, that is flame ionization detector, which is not commonly used in GC. Coming to argon, argon is a carrier gas for TCD, thermal conductivity detector. Coming to hydrogen, hydrogen can be used as a carrier gas for capillary conduction, as well as it can be used along with oxygen for the combustion in FIDs, that is flame ionization detectors. Because these are, although cheap, these are explosive, so it should be handled carefully. Coming to air, the normal air can be used as a combustion gas for various type of detector. It is cheap and readily available. Right. Coming to a combination of argon and methane, it can be used as a makeup and a carrier gas for ECD detectors. For ECD columns, it has better linearity and specificity than nitrogen. But the thing is, it is having a poor detection limit. That is the drawback of a mixture of argon and methane. Right. So what is this gas chromatography or GC? Let's study about the basic of this GC. So what is the purpose? It is to separate, identify and quantify individual chemicals in a complex mixture. Right. So here what we want, instead of a mobile phase, we will be using a carrier gas. And the carrier gas is considered as a mobile phase here. Okay. It can be hydrogen, nitrogen or helium, etc. So these are used as a carrier gases. What is the requirement of the sample? The sample should be volatile in nature, but it should not undergo any type of thermal decomposition. But it should be volatile in nature. Okay. Now coming to the main component, we should inject this or introduce the sample, right? So that's why we are using an inlet. So this inlet will introduce and vaporize the sample. Now this vaporized or volatile form of sample will enter into the column. Now it can be split or splitless. SSL is mainly for splitless columns, splitless inlets. So this is a common type of inlet which is used for optimizing the analysis. Now there are many other type of inlets also, such as PTV. PTV is a programmable temperature vaporizer. So here we can rapidly increase the temperature or rapidly decrease the temperature. That's why it is PTV. Now, coming to COC. COC is cool-on column. In cool-on column, we are directly injecting that into the column at low temperatures. Right. Coming to MMI. MMI means multi-mode inlet. In multi-mode inlet, we will get features of the rapid temperature control as well as splitlets injection inlets. So these are the main feature of MMI, that is multi-mode inlet. So here we will be using a column, right? So this column is for the separation of compounds based upon the interaction with the stationary phase. Coming to the structure, it will consist of fused silica tube with polyimide coating. Right. The stationary phase, it will depend. The stationary phase can be of various types depending upon the polarity as well as what type of sample we need to separate. It will differ. Now, separation principle is like dissolves in like, which means polar analytes preferably will dissolve in polar phases, while a non-polar always prefer the non-polar phases. Now coming to the temperature control, as we said, it should be always volatile in nature. So the temperature should be controlled. If there is a drop in the temperature, there are chances that the sample can condense, right? So which is not desirable. So we are always having a temperature control. Oven ramps will help separate the compounds by altering the partitioning, right? So coming to the detectors. Detectors is for identification as well as quantify the compounds as they exit the column. What are the common detectors which are used in GC or gas chromatography? It is FID, that is flame ionization detector, mainly specific for hydrocarbons. Then comes TCD, that is thermal conductivity detector. These can detect all kinds of organic compounds except for the carrier gas that we are using. Now coming to the ECD, NPD and FPD, these are specific detectors. Like NPD is mainly for nitrogen and phosphorus containing compounds, like that. Coming to MS, this is GCMS, gas chromatography mass spectrometry. This will help to identify the unknown compound also. For unknown identification also, By using the GC, we will separate the mixture. By using the MS, we will identify the compound. This can be combined. Now, what is the output that we are getting? That is a chromatogram. So, coming to the output, we will, in the x-axis, we will get retention time, and the y-axis, we will get detector response. Now, retention time means what? It is the time that the compound takes to travel through the system, or the time the compound takes to reach the detector. That is the retention time. Now, here we can also use this chromatogram for quantitative analysis by using calibration curves. Like by using a standard, we will draw the calibration curve and identify the concentration of the compounds. In which of the following separation technique, the separation is based upon the net charge of the molecule? Is it affinity chromatography, ion exchange chromatography, molecular exclusion chromatography, or GC, gas chromatography? Yes, it is ion exchange chromatography, because in the question itself they have given net charge of the molecule, right? So, ion exchange chromatography, what is the principle? It is based upon, the separation is based upon the net charge of the molecule. The stationary phase will be charge group. It can be either cationic exchanger or anionic exchanger. Now, coming to the sample interaction, ions in the sample interact with the oppositely charged group, which is present in the stationary phase. Right. Now, coming to the elution, in elution, by varying the pH as well as varying the ionic strength of the element, the ions can be separately eluted as per the requirement. Right. The separation and so separated based upon the net charge which is present upon the compound or the molecule. That is ion exchange chromatography. Hope you enjoy the video. Thank you.